Lecture 9. As we know, phenotype equals genotype plus environment. But how does genotype influence phenotype? The central dogma of biology addresses this question. The central dogma explains how information encoded in the DNA, the genotype, is transcribed into RNA, which can be spliced and then translated into protein, which gives rise to the phenotype. DNA encodes the information, but the proteins carry out the functions of living organisms. Schematically, we can see how double-stranded DNA is transcribed into messenger RNA, which is read 3 nucleotides at a time to give rise to an amino acid sequence that folds into a functional protein. Over the next few slides, I'm going to quickly review the processes of transcription, splicing, and translation. The point is to gain insight into how mutations or changes in DNA sequence can give rise to changes in mRNA sequence and then changes in protein sequence and function. The first step is that the DNA of a gene is used as a template to synthesize a complementary mRNA that captures the information in the DNA. One strand of the DNA acts as a template for a complementary strand of RNA to be synthesized by RNA polymerase. This process is regulated by transcription factors that either increase or decrease the rate of transcription, resulting in more or less mRNA. This transcriptional gene regulation is mediated by transcription factor activators or repressors that physically bind to regulatory sites on the DNA, such as promoters, enhancers, or silencers. By binding these DNA elements, the transcription factors can activate or repress RNA polymerase. In eukaryotes, the primary transcript or pre-mRNA contains regions called introns that don't encode for protein, and they need to be spliced out by RNA splicing. The exons are regions that encode for protein. So we can see the DNA of a gene is transcribed into a pre-mRNA or primary transcript that contains introns and exons. The introns are spliced out to give rise to the mature mRNA that is translated into protein. The splicing reaction is catalyzed by the spliceosome. The spliceosome recognizes the 5' and 3' junctional sequences of an intron, the GU on the 5' end, and then the AG on the 3' end of the intron are in particular necessary. The spliceosome can then remove the intron and connect the exons together. The mature mRNA is translated into a protein sequence by the ribosome. The ribosome uses the mRNA as a template to synthesize the protein. The individual amino acids are attached to tRNA molecules which possess a 3-nucleotide anticodon that recognizes each 3-nucleotide codon on the mRNA. Here we can see the translation process happen with the ribosome initiating translation at the AUG start codon of the mRNA, and then adding amino acids brought in by tRNA for each successive codon in these steps until the ribosome encounters a terminator codon and stops. Right here. During translation, the amino acids are linked together by peptide bonds to form a polypeptide chain or protein. There is a direct relationship between the DNA and mRNA sequence and the protein sequence. The translation manual is provided by the genetic code. The key insight was that the mRNA sequence is read by triplets which form a codon. Each codon specifies an amino acid or one of the 3 terminator codons. The genetic code is highly conserved in all living organisms. And here is the magnificent genetic code. One thing to note is that the code is degenerate. Most amino acids are specified by more than one codon. This can be seen by the fact that there are 20 amino acids, but the codon is a triplet of nucleotides. As a result, there are 4 times 4 times 4, or 4 to the third, which equals 64 possible codons encoding for 20 amino acids and the 3 terminator codons. Thus, most amino acids have 2 or more corresponding codons. Proteins are polymers of amino acids. They are connected by peptide bonds that are formed during the translation process. The sequence of a protein determines its structure and function. Proteins perform the functions of living organisms. Part A of Assignment 9 addresses the following question. Pretty much every cell in your body has the same genotype or DNA sequence. Then how do the different cell types arise? How does a neuron differ from a liver cell? Create a Google document titled Assignment 9 and answer this question. The second part of Assignment 9 is to add more information to Question 7 of your term paper. Already in Question 4, you have identified 3 or more genes that contribute or influence your complex trait. Now, I want you to identify alleles in those 3 genes. What are the major alleles of these genes? You can Google the specific gene names followed by the word allele or alleles. I want you to find out as much information about these alleles as possible. In particular, I want to see if you can determine what specific mutation or mutations give rise to these alleles and how those mutations may alter normal wild-type protein functioning, influencing your complex trait. In summary, the central dogma shows how genotype can influence or contribute to phenotype. DNA is transcribed into mRNA, which is spliced and then translated into protein. The genetic code translates the information in the DNA or RNA sequence into protein sequence. Mutations in the DNA can affect this process so that a change in the DNA sequence can result in a change in the RNA sequence into a change in protein sequence, which can change function and the phenotype. And that will be the subject of Lecture 10.